And I'm Dulmini Nayakar, Research Fellow of Combivet Russia. And then today, the guest lecture is about blood, sweat, and tears, extracellular vesicles, and coagulation. And the speaker is Dr. Rienk Nieuwland. Let me take a few minutes to give a brief explanation about him. He's the chair. He chairs the Amsterdam Vesicle Center of the Amsterdam University Medical Center. He focuses on isolation, detection, functional characterization, and standardization of measurements on extracellular vesicles for biomarker exploration. Moreover, he coordinates EURAMET projects MedVest and MedVest 2 aim to standardize concentration measurements of EVs. Dr. Nieuwland chairs the Scientific Standardization Committee on Vascular Biology of the International Society on Thrombosis and Hemostasis, and co-chairs the Rigor and Standardization Committee of the International Society on Extracellular Vesicles. He is also an executive board member and chairs the Educational Committee and Blood EV Task Force, and also co-founder and the member of the Extracellular Vesicle Flow Cytometry Working Group. Lastly, he is the Vice President of the Netherlands Society for Extracellular Vesicles and co-organizes a recently founded working group on ISEV and the European Liquid Biopsy Society. So today, he's going to talk about blood, sweat, and tears, extracellular vesicles, and coagulation. Over to you, Dr. Nieuwland.
Thank you for this kind invitation. I will try to share my screen again. You never know whether that works. Yes, can you also see my screen?
Yes, we can see.
Okay.
Right.
Yeah, so the title of my presentation today is Blood, Sweat and Tears, Extracellular Vesicles and Coagulation. First, I would like to introduce our research group. We have a research group in Amsterdam, the Amsterdam University Medical Center, that we now call, since 1 or 2 years, the Amsterdam Vesicle Center. And actually, this group of people is quite unique because we have people that are from 2 different departments that are collaborating. So we have people with more chemical background, so people working in the group of experimental clinical chemistry. And we have people with a background in physics, and they are from the Department of Biomedical Engineering and Physics. And what the overall goal or focus of our research is that we are developing novel techniques, novel methods to study the presence of EVs and their function, especially in clinical samples. We're working in a large university hospital. And the ultimate goal of all our studies aim to explore the presence of extracellular vesicles in human body fluids as biomarkers of health and disease. Just checking whether I can somehow make a pointer. What's the like this? Okay. So, and our expertise, I just try to summarize this slide, is very broad. We have knowledge about how to collect blood and how to prepare plasma and serum, but we also have people in our group who are developing software and people who are even developing hardware components for flow cytometry. So we have a very broad group of people that work really very translational. Perhaps one of you may have seen this paper that came out very recently, written by a journalist, Alison Abbott. She's a journalist for Nature, and she interviewed, I think, quite many people who are now quite active in the vesicle field. I was very happy that I was also invited to tell a few things about what is going on the standardization of EV research. And as you can see of the title, the title is the cell-to-cell postal service that could help to treat disease. And it's the cell-to-cell postal service that everybody in the whole world seems to be working on, the role of extracellular vesicles in intercellular communication. Well, I still have doubts about it, and that's more or less what my talk is about. So in this nice Nature paper that was written by Alison Abbott, she put one quote, and that's that she put really big in the text, and actually that's my quote. And my quote was, "I've never seen conclusive evidence that extracellular vesicles do anything in vivo." And when I saw this, I thought, oh no, have I really said it? But I think I did. And in my talk now, I will try to convince you that I'm pretty sure they do something in vivo, but I'm unsure, to be honest, whether that is related to intercellular communication. Maybe it is, maybe it isn't, but I have some other ideas, and also people like Johannes Thaler and Carla who are online, I think, share these ideas with me. So I would like to speculate or discuss with you what is the function of extracellular vesicles in vivo. And this is a peeing male dog. And I put this slide here because for me as a biologist, it is still hard to understand why vesicles, abundantly present in saliva, urine, and all sorts of body fluids, why would such vesicles be all involved in intercellular communication? I really have no clue. I cannot explain it, also not as a biologist. So what is the function of all these vesicles that are in those body fluids? So my talk will really be about vesicles and their role in physiology, and I hope in this talk that I can convince you that I think the primary function, or one of the main functions of these vesicles, is that they may be able to protect you. In fact, I hope to convince you that as you're sitting here and listening to this talk, I think you wouldn't be sitting there alive if you would not have your vesicles. I think that's how important they are to protect you. Let's see whether I can convince you. You can judge in the end. So this is my favorite model, a bleeding finger. I hope to show you why I think this is an interesting model. So if you have a bleeding wound, what your body does, it tries to close the gap, the hole, as soon as possible. Why? Because you want to restore the integrity of your body. Why do you want to do that? I think the main reason is that you would like to prevent microorganisms, bacteria, viruses, whatever, to enter your blood. Because if they do, you can develop infection, sepsis, and then you die. So if you have a hole somewhere in your body, the main goal of the body is to close this hole as soon as possible. I think this really helps you to survive. Now, to explain a bit what is going on if you have a bleeding finger or a bleeding wound, I have a few slides that if you get lost in this background, it's not a problem, because in the end I will also show it in a different way. But this is just to show you in a sense what is happening. So what is happening is, if this is the vessel wall and you have a bleeding wound, the vessel wall is damaged. I just did not put an endothelial cell here. The subendothelium becomes exposed. The next step is that your blood is flowing here, and the first thing that will happen is that blood platelets will immediately adhere to these collagen fibers and all of other fibers that are exposed. And these blood platelets, so cells present in your blood, what they will do is they will adhere to each other and they will loosely close the wound. But you should imagine this as sort of bricks. If you put the bricks on top of each other, it's not a very stable construction. So to make this a stable construction, a stable cloth, you need some mortar. In this mortar, the compound that glues all these platelets together, these are these red strands. I indicated, I know it's very schematic, but these are the fibrin strands that are being made. And this makes a sort of meshwork, a network that glues the platelet together. And together you have a thrombus that closes the wound. You also can say this is a wound crust. Now, my talk will be about the process that generates these fibrin fibers. Now, what do you need to make these fibrin fibers? Well, you need a couple of compounds, and these compounds have in common that they're all present in large amounts within your in your blood when you're normal humans. So if you do not have any problems, this is all present in your blood. So you need calcium ions, coagulation factors, the substrate fibrinogen, that's a soluble protein, and that's converted to fibrin, which is insoluble, and you need a membrane surface. Now, why does your blood not clot? Well, if you look at coagulation, and this are a number of coagulation factors, and I know this is a very simplified scheme and you don't have to remember it at all, but what happens in this scheme is that all these components are present within your blood, and the final active enzyme of the coagulation cascade, which is thrombin or coagulation factor 2A, converts soluble fibrinogen into insoluble fibrin, and these are the strands gluing the platelets together. Now, there is one compound here that I did not draw, and that's the compound that is not present in your blood under normal physiological conditions. And that compound is tissue factor. And tissue factor is a protein, I will come to that in a minute, that is present outside your blood. So, and what happens is that if tissue factor present outside the blood vessels contacts the blood, so you have a wound, so the blood can ooze out of the wound, what happens then is tissue factor will bind factor 7. It catalyzes the activation of factor 7A, factor 10 is formed, thrombin is formed, and you get fibrin formed. This is a very fast reaction. You don't have to remember all this, but please remember that tissue factor binds factor 7, you get 7A, and in the end, you get fibrin formation. Now, tissue factor is a very interesting protein. Tissue factor is a very abundant protein expressed by many types of cells. It's a transmembrane receptor, so it's not present as a real soluble protein normally, but it's present as a receptor on cells or on vesicles. I will show you in a minute. And in the normal conditions, as you are listening to this talk, this protein is not present within your blood. I think this is something that you really have to remember, absent within your blood. When you're ill, for example, if you have cancer or you develop sepsis, this protein can be present within your blood. But I think I removed all slides. I also have many slides about tissue factor pathological conditions. I will not tell about that today. It's just that you know that for 30, 40 years, we already know that in pathological condition, tissue factor can be present within your blood. And people think there's associated with extracellular vesicles. Now, tissue factor, transmembrane protein, not present in your blood, but the ligand is a coagulation protein, coagulation factor, factor 7, and that is present in your blood. So the ligand and the receptor have a different location in your body. So the receptor is present outside the blood, and the ligand is present inside the blood. Now, as I said before, when you have a bleeding wound, blood goes out of the vasculature, and in that case, blood containing factor 7 meets tissue factor, and coagulation can be initiated. And this complex of tissue factor together with factor 7, 7A is called the extrinsic tenacious complex in some studies. Okay, now I just before this talk I made a few slides because I thought maybe you get lost here, but it's really simple. With this I indicate the tissues and the organs are full of tissue factor. This is the vessel wall, and within the blood, this is the blood vessel, and here you see the wall. Within the blood, you have factor 7. So it's just to illustrate that the receptor and the ligand have a different location, and when the blood leaves the blood vessel, it can contact this tissue factor, which is really present everywhere. That's important to keep in mind. So now coming back to this bleeding finger, of course, what I said is what your body would like to do is to close this wound as soon as possible. Now it becomes interesting because if you have this wound, one of the processes, I hope that I just showed you, that will be triggered is coagulation. Coagulation is really very complex, and I think everything I showed a few slides ago you can forget because it's not so important for this talk to understand. You end up with closing a wound, can be a wound crust or whatever. But, and this is in a sense where my talk is about, what many people do when they have a bleeding wound, or a small bleeding wound, they start licking the wound. And what you do is when you lick a wound, you expose the blood to saliva. And that's not only something that humans do, but that's also something that animals also lick their wound. And years ago, a postdoc in our lab came with the question, why do we lick a wound? And I said, I have really no clue. But we published a paper where we did what I always call some Friday afternoon experiments. And what we showed actually in a set of very simple experiments, that if you collect normal human saliva, so really these are experiments like spitting in a tube, you can very easily demonstrate that normal human saliva contains vesicles, and these vesicles, a large number of these vesicles, large concentration of these vesicles exposes tissue factor. Tissue factor, what I said, the protein that initiates blood clotting. And these are just two of the experiments we did to show that the tissue factor antigen is associated with vesicles. For example, if you have cell-free saliva, so first you remove the cells at a low speed centrifugation step, and then you centrifuge that cell-free saliva at 18,000 G or at ultracentrifugation speed. And this is for data from 3 donors. What you see is that for all donors, we see considerable amounts of tissue factor antigen that we can pellet. And here you see that the tissue factor antigen is really associated with a vesicle in this saliva. Now, the proof of the pudding is, of course, do these vesicles really trigger blood clotting? And the answer is yes. I forgot to explain this on the previous slides, and I noticed, but when you have the blood fluid, plasma, and probably many of you will be familiar with it, it's a clear yellow fluid. It's really a fluid. But when the plasma clots, it becomes a gel and it becomes very turbid. And this conversion from a clear fluid, to a turbid gel can be very easily measured in vitro on an ELISA plate. That's what we do. So you can just measure the light that's falling through the plasma at optical density of 5 nanometer, and the experiment you do at 37 degrees. And what we do is here, at T0, on the x-axis you see the time, so this is 0 and this is 1 hour. This is the optical density on the y-axis. At T0 we add plasma, normal human plasma, which we recalcify so it can clot, it needs calcium. And what you see is that nothing happens. This plasma does not clot. If, however, you add saliva, if you mix a little bit of saliva with this plasma, or you add the vesicles from saliva, you can see that here the plasma goes from a clear fluid to a turbid suspension, and that's in fact a reaction that's indicated here, and you see this goes really, really very fast. Meaning that if you mix blood with saliva or plasma with saliva, you get coagulation initiation, and it's very fast. So, in fact, what we did is we collected normal human saliva and compared it to the ability of a snake venom, and that snake venom is an enzyme that's called reptilase. Reptilase directly converts fibrinogen into fibrin, so it directly lets the plasma clot. And actually, if you do a head-to-head comparison between saliva, or the vesicles in saliva, and reptilase from the snake, they're about equally active, meaning that your saliva has as much clotting activity ability to trigger blood clotting as the fastest working snake venom. So it's really very active. And perhaps surprising, perhaps not surprising, but with medical students, we never published it, but we also collected sweat and we also collected tear fluid. And you can do exactly the same experiments as we did with saliva. And it's very easy to see that also these fluids they trigger blood clotting. It can be completely attributed to the present vesicles exposing tissue factor in these fluids. And here, actually, what you see here is on the lab bench. I'm not sure whether it's really allowed, but we were cutting onions so that the girls would start crying, or the boys, and we would collect the tears and then do our experiments. Actually, it's not so easy to collect tears, but it's a different story. So, what we now show is that this was the tissue. Tissue contains tissue factor, but if you look at the body fluids like tears, like saliva, and then some other body fluids, they contain vesicles exposing tissue factor. And if you add them to the blood, they greatly accelerate the clotting of the blood. Now, then, I don't know, maybe it's already 4 or 5 years ago, I forgot, but then Johannes Staller, who's now also online, came to me. Johannes is an Austrian pediatrician, clinician, hematologist, and he told me that during his night shifts, he is always looking into this old literature about all the experiments that have been forgotten. And one of the very interesting findings that he did was that he came up with manuscripts from 1934 to 1935 showing that people have been studying the effects of mother milk and adding gauzes soaked in mother milk to wounds. And they showed, and I think that's quite remarkable, that if you have a bleeding wound, and they studied this in patients with hemophilia, and if time allows, I have a few slides on that in the end, but there are children, mainly boys, with a bleeding tendency, and the bleeding, the external bleeding cannot be stopped. And what they did in those days, they had gauzes soaked in different fluids, for example, milk from humans, but also milk from cows, for example. And they looked whether these fluids are able to stop the bleeding. And then, this is a quote from Landsman, translated, I guess, but no other animal milk has such blessed hemostatic properties as human milk, as first shown and described by the Viennese pediatrician Alphonse Soleil in 1934. Put a tamponade soaked with human milk and press it on the bleeding spot. So what they showed is that the addition of milk on a tamponade or gauze on a wound stopped the bleeding. And Johannes, shown here on the left with, I think, the second wife of Alphonse Soleil, came to me with the question, how does human milk stop the bleeding, and is there any role for vesicles? And this is the work mainly of Jung and also Johannes and many other people, but I think they did most of the work that we published a few years ago, where we showed that human milk triggers coagulation, just as saliva, just as tear fluid, etc., by containing tissue factor exposing extracellular vesicles. And I think what for me was perhaps the most surprising finding was, here you see milk dilutions, normal human milk dilutions. Here you see the buffer, and here you see the plasma clotting time. So we used exactly the same test. We measured up to 1 hour. If you just had buffer, the plasma will not clot, so the clotting time is 1 hour or longer. But if you add increasing concentrations of milk, what you can see is that the clotting time decreases dramatically. And in fact, if you dilute it, if I remember correctly, mother milk up to 4,000 times, we were still able to measure a significant clotting, a shortening of the clotting time. So the biological activity of vesicles in normal human milk to trigger blood clotting, as in saliva, is extremely, extremely strong. In fact, in milk, it's even stronger than it is for saliva. And these are just a few experiments we did to show that the tissue factor antigen and activity are really associated with vesicles. And I think for those of you who are working with vesicles, we use size exclusion chromatography, cephalose 2B, to prepare 26 fractions, as we described earlier, also for plasma. And as you can see, if we blot these 26 fractions from milk for tissue factor antigen, you can see that we see only tissue factor in fraction 8, 9, and 10. So these fractions contain the tissue factor antigen. And what you can also see is that these fractions also contain the coagulant activity of the milk, and we could block this activity with antibodies against human tissue factor or human factor 7. So I think this quite convincingly shows there's a fraction in human milk, very likely vesicles, that expose tissue factor and can trigger blood clotting. So this is a graphical abstract that Jung Hu prepared, and I think it very elegantly summarizes the main finding of this manuscript, and it is that normal human milk contains vesicles with exposing tissue factor. Tissue factor binds factor 7, as I told you, present in blood, and it makes a complex. This complex activates factor 10, 10A is formed, thrombin is activated, is formed, and fibrin is formed. So this is a very nice, elegant pathway. Now, actually, what we showed, I think a year later, is that we were wrong, because what we discovered is that actually the vesicles in the human body fluids do not only expose tissue factor, but they also expose 7A. So meaning that the complex of tissue factor is the tissue factor on these vesicles in body fluids other than blood. This tissue factor is already associated with factor 7 and activated factor 7. And just to summarize this graphically, so if you look at the body fluids like saliva and milk and tear fluid, they not only contain extracellular vesicles exposing tissue factor, but they actually expose the extrinsic tenase complex. So they are very potent and they can very fastly activate the coagulation and thus promote hemostasis. Now, there was an interesting observation that was done already in the 34, 35, and that was that human milk stopped bleeding, but in contrast, bovine milk did not stop bleeding. And this was something that sort of we find very intriguing, because I somehow would expect that bovine milk and human milk and goat milk and whatever, they all have the same properties. Now, there can, of course, be two explanations. One explanation, I think that's the most likely one, is that there is no reaction between bovine tissue factor and human factor 7. I think that mechanistically would explain why it doesn't work. The other explanation, which I thought is much less likely, is that bovine milk contains vesicles, but it does not contain vesicles exposing tissue factor or tissue factor and factor 7. So Jung Hu, together with Shi Hao, spent a day in a farm where they collected several body fluids from cows. And they had actually a quite surprising result. So here you see human milk, Western blot from 3 donors, and bovine milk plotted for anti-tissue factor. It's of course an antibody against human tissue factor. Well, that's what we indeed see, but we don't see anything here. So of course it can be that bovine tissue factor is not identified by antibodies, by an antibody against human tissue factor. But we also did simple coagulation assays. So again, on the y-axis you have the plasma clotting time. If the bar is high, it's 1 hour longer, there's no clotting. And if the bar is low, there's clotting activity. Now what you can see is that if you take human plasma in these three conditions, this is the blank, so there's no clotting. If you add bovine milk, what you see is that nothing happens. If you add human milk, you can see that, as expected, there's a clear shortening of the clotting time. Again, if you add bovine milk to human plasma, it can be the lack of cross-reactivity. But here we had bovine plasma to which we added bovine milk. And as you can see, there's absolutely no shortening of the clotting time, which is very surprising. So it seems there's no disinfectant activity. And in fact, if we add human milk to bovine plasma, you do see a shortening of the clotting time, meaning that a tissue factor present in human milk, or tissue factor 7A, can really activate the clotting system in bovine plasma. So it seems that bovine milk does not have any detectable tissue factor activity. Now we became curious. I'm a biologist, so if it's only one fluid, so I was curious. How about bovine saliva? Because we do know that animals, also cows, are licking their wounds. So we also collected saliva, and I can tell you it's the only green saliva samples we have in our fridge in Amsterdam, because they're really green. It was quite hard actually to do that. But what we could clearly see is that if you have bovine plasma and you add bovine saliva, that is really a shortening of the clotting time. So we do not have specific antibodies against bovine tissue factor, but I think it's very, very likely that in contrast to bovine milk, that bovine saliva also contains vesicles exposing tissue factor. At least they promote coagulation. So to summarize, as I told you, in the normal physiological conditions, human blood absolutely does not contain any tissue factor. If you look at human milk, human saliva, sweat, urine, tears, they all contain tissue factor activity and likely tissue factor 7A activity. If we look in bovine milk, we cannot detect any tissue factor activity, and in saliva, I think it's present. Again, keep in mind, it's a huge difference. We could dilute human milk 4,000-fold, and there was triggering of coagulation. There was a shortening of the clotting time. Here, we cannot detect any activity. We have no idea. We cannot explain this easily because we did not look in longitudinally, so we did not look in the first milk like colostrum. Perhaps if you have non-domesticated cows, it's different. Many questions that you can ask, but the effect as such is very different. So coming to challenges and question of the relevance, it seems that there are functional differences of milk vesicles between the mammals. There are functional differences in body fluid-derived vesicles, even between in one animal species. I also found that quite surprising. And if you look at our findings, is this of any relevance for human health applications? Well, we used it for 2 clinical applications. We used it for the screening of human donor milk, which is pasteurized to kill the bacteria there. We've used human milk tissue factor activity as a reference material in the laboratory comparison study organized by ICH. And we have done a couple of studies, but that's more pathology, where we try to predict the risk of developing venous thrombosis in cancer patients. And I will not show this. I think for time's sake, I will skip the examples unless you're very interested in that. I think it's not so relevant to highlight that for understanding the story. What I would like to show you is the last few slides. Coming back to this quote, I perhaps never should have said, but how about conclusive evidence that extracellular vesicles do anything in vivo? Well, I showed you this. We do know now that if you look at these body fluids, they seem to have tissue factor and factor 7. So I'm talking about saliva, milk, tears, etc. So they have a greater activity. It's very active. Now I come back to an experiment that's quite recently was published in an experiment by Johannes Staal and Mercala, I think. And I would like to show it to you because I think for me, this is one, it's still indirect evidence, but I think it's pretty convincing and it comes very close to what vesicles may be doing in vivo. Likely they have many more functions, but this is how I see it. So if you have patients with hemophilia A and B, inherited bleeding disorder, these patients have reduced activity of coagulation factor 8 or 9. If you are lost where they are, I'll show you that in a minute again, because I think if you're not familiar with clotting, you're lost immediately. But these patients present with spontaneous joint bleedings. They have bleedings in muscles and soft tissues. But the important thing is they have no or very rarely bleedings in their oral cavity. So they not or very rarely bleed in their mouth. Now, why is this? Now, if you think about the blood of hemophilia A patients, this blood is lacking factor 8. If you think about their saliva, if you are deficient of factor 8, you expect that the saliva of these patients with regard to vesicles will be sort of normal. I would expect, or we would expect, Johannes would expect that the saliva from these patients will contain normal vesicles, meaning vesicles exposing tissue factor together with factor 7A. And the question they asked themselves is, can saliva likely containing vesicles with tissue factor 7A from hemophilia A patients, can it restore the ability of factor 8 deficient blood to clot? So what they did is actually, I think, very simple, very elegant. They collected blood and saliva from hemophilia A patients. Keep in mind, these patients have a bleeding disorder. Their blood does not clot normally. The question is, what happens if we add saliva from a patient to its own blood? And actually what you would expect is that in that case, there would be a normal ability factor 8 deficient blood to clot. So what you do is coming to the scheme. So if the saliva is an important source for tissue factor and factor 7 also in these patients, you would expect that mixing saliva containing vesicles exposing tissue factor 7A, it would trigger fibrin formation. These patients are lacking factor 8, but as you can see, you can completely bypass this part of the coagulation cascade when you add sufficient tissue factor in 7A. This factor is completely not needed. I hope I explained this properly, and if not, I hope Johannes in the end will correct me. And this is the experiment what they did, and I still think it's a really brilliant experiment. They mixed saliva and plasma from patients with factor 8 deficiency. And what you can see is that when you add increasing concentration of saliva, and even urine, what you can see is that it, as in the other figures, it shortens the clotting time. And I think this very elegantly shows that this may be the underlying explanation, although again, still not completely direct evidence, but it may be the underlying explanation that patients with hemophilia A do not have mucosal bleedings. So, what I hope you have learned today from my talk is that normal human body fluids contain extracellular vesicles and they abundantly expose tissue factor 7A. And this complex triggers blood clotting. So these vesicles promote coagulation and hemostasis, so they promote wound healing. I think it's very important, as I said, because you don't want any infection. The presence may explain perhaps our reflex of licking the wound. We have as yet unexplained differences between mammals and body fluids. Much more work has been done there. Regarding the function of these vesicles in vivo, I think in general it has to be explored. But my hypothesis would be that what I would call normal vesicles, or vesicles that are present in normal human saliva, exposing tissue factor 7A, I think they may, in principle, play a role at the interface, and that keep in mind your mouth has lots of pathogenic bacteria. So it is important at those spots that you accelerate hemostasis as much as you can, thereby reducing the risk of infection. Of course, it's an hypothesis, but still, to me, it makes a lot of sense. So my take-home message is that I hope that I have convinced you that if you think about extracellular vesicles, that I think very likely they do play a role in protecting your body by promoting hemostasis and by trying to keep the nasty germs outside your body. So, and these are the people I really would acknowledge. Of course, Johannes Stahler, Chi, who did many experiments together with Jung is shown here. René Bergman also did a lot of work, especially on the saliva vesicles. And Carla, who is a postdoc, joined the group recently of Johannes Stahler. So, thank you for your attention. I hope you managed to follow it and try to explain it to the best of my abilities, but I'm perhaps not the best explanator, but let's see with your questions what you got from it. So thank you very much for your attention.
Thank you, Dr. Nieuwland. So now the floor is open for the questions.
Adi Reza has a question.
Yeah, Dulmini, I'm trying to find out how can I put my hands up in here. Yeah.
It's in the reactants.
All programs are different.
Yeah. Is it okay, Dulmini? Can I?
Yes, please go ahead.
Okay. Thank you very much. It was very interesting, especially when you showed, although I saw mainly the effect of concentration when you were using the whole saliva. So I don't know if you have also done those experiments with EVs, and if that is there.
Yes, you can also do that. It's these experiments are really very easy to perform. So we have all sorts of experiment, dose response curve, etc. Then you see essentially the same.
Yeah, that shows that it is really EV effect, or it is a real effect that you can also measure it and bring that. One thing I was wondering was the slides, maybe it was in those slides that you were having it about the pasteurized milk. I didn't understand. Do the pasteurized milk, do they have the same effect or they don't have the same effect?
Yes, I can show you that if you want, then I have to share my screen again. Is that allowed?
Yes,
Sure.
that's okay.
Okay. So let's see whether I can now. So let me see, I have to go scroll down a bit. Yeah, I know Estonia is a very dairy minded country. You have a lot of dairy cattle, so really this is indeed interesting for you.
We are producing the highest amount of milk per dairy cow.
Yeah, so let's go here. Yeah, so this is a study we published, I think, when was it? Quite recently, 2022. So we have I'm working in a, as I said, in a large university hospital, and human donor milk is collected for patient care, and of course this milk can contain bacteria. So this milk is pasteurized, and what we did is, I think, really simple. We used the plasma clotting assay as a readout to test the effect of pasteurization on the tissue factor activity that's associated with the vesicles. So what is this? You see, it should be about 30 dots. This is milk from 30 women, normal healthy women, that was either left untreated, so this is not pasteurized. So again, this is on the y-axis, the plasma clotting time 1 hour or longer. If you add, I think this is 1 or 5% of volume that was added. If you add milk from 30 donors untreated, what you see is that the clotting time goes from about 3,600. That's actually here shown in the buffer control. It's really 1 hour. But if you add 1 or 5% milk of 30 donors, all untreated, not pasteurized, you can see that the clotting time shortens dramatically. Now, I think this is Holder pasteurization. This is the normal pasteurization procedure that is done. I really forgot the details here. But what you can see is that there's quite a big effect of Holder pasteurization compared to untreated. This is high temperature, short, I forgot what this T is.
Short time.
Is it? Okay.
Short time.
Short time. Okay. So what you see is that also here it has quite a dramatic effect compared to untreated. Thus, I really forgot. I don't know, but this is also some heating procedure. But what you see is that the 3 procedures of milk pasteurization, where the milk is heated, they all have quite a big effect on the tissue factor activity. So they deteriorate, they damage the tissue factor activity. If you look at the treatment with ultraviolet C or with, this was, I forget, high pressure pasteurization, I think also. So in these 2 procedures, there's no heating involved. What you can see is that the tissue factor activity is much better retained compared to untreated than when the pasteurization methods, when the milk was treated. So the question, of course, is the tissue factor activity, is it a good readout to screen the milk for potential, let's say, the well-being of the infant when it consumes the milk? That's difficult. On the other hand, it's an easy, simple, fast way of screening milk, and it's very reproducible. So and of course it was very clear that as soon as you start heating the milk, the tissue factor activity is reduced very strongly.
And EV activity, because I think there is a debate out there about the EVs in the milk, that if you pasteurize the milk, do the EV function goes away or would it stay there? And I think there are several different school of thoughts there. Some are saying no, maybe it is depending on what kind of function you're looking. Some people look at it and say it stays, some other. But in your case, even you say depending on which kind of pasteurization you take.
It is very clear from our studies is that compared to untreated milk, that in the 3 pasteurization ways of heating the milk, in all 3 we see that the tissue factor activity is strongly reduced. And when you compare it to UVC light or high pressure, there's hardly any effect. So yeah, I don't know whether it's the tissue factor, whether it's the vesicles that are damaged by heating, but there is an effect, yes. At least when you look at tissue factor coagulant activity.
Thank you.
Yes, get that.
Thank you, Professor Rienk. Yeah, I have a couple of questions. I don't know if you can go, if we can see your slide again, slide number 39. Can we have a look?
Which slide would you like to see again?
Slide 39.
39. Ooh,
yeah.
That's let me see what I can do. Can you see my screen now or not?
No, not yet.
No, not yet. Okay. Give me a second. How can I get out of this? Oh, I'm sorry.
It's about like
yeah, I know. I'm just doing.
There's no detection of this TF antigen and no activity in bovine milk. My question.
Yeah, give me a second. This is
39.
Yeah, I know, but I would like to put it bigger. 39. Yeah, please.
Yeah. Here, like you said, no detection of TF antigen activity of bovine milk. My question here: what kind of milk are used here? Like when you say bovine milk, like raw milk or
Yeah,
treated?
this is just raw milk. Yeah.
Okay, and maybe in that case, and we don't know also how you purified this sample. Like, what was the procedure? Like, sake or what is in whole scenario?
Sorry, here's completely nothing. This is just two fluids mixed.
Just like
So we just mixed. Sorry, we just mixed human plasma, bovine plasma, with 1 or 5 or 10% human milk or bovine milk, etc.
Yes, yeah, I understood that. But how it was prepared, this milk?
Yeah,
Do you have the information? Like, I mean, how do you prepare it?
Yeah,
I think I can jump in because
yeah,
Yes, please go ahead.
Yeah, there is as a publication in Journal of Extracellular Vesicles from Sonefeld et al. And they suggest a protocol how to prepare milk. And it was centrifuged 2 times, I think at 3000 G, and always the cream there. So if you centrifuge the milk, you get a cream layer, which is only the lipids, and you remove the cream, and then you centrifuge it 2 times. And this protocol was shown to quite well preserve the vesicles, and you get rid of cellular debris and so on. And this protocol, it's also in our publication cited. It's published in JEF, and I think it's an accepted method how to store the milk samples.
Okay, yeah. So it's only centrifugated. Yeah. Hello, can you hear me?
Yes, go ahead.
Maybe this is the reason. Maybe we didn't get this our like bovine milk. Maybe they are not presenting their this TF antigen because they are blocked in this. Maybe in cases if it's only centrifugated, they are not. There won't be any binding site. This ligand and receptor reaction, so this clotting will be dropped. Maybe that's my thought. Yeah, this is my suggestion.
Sorry, I do not understand what you say. Sorry.
My question, I think if in this procedure, if we only use centrifugation, you are not exposing the TF antigen in bovine EVs because there is everything, maybe the corona, everything is like totally blocked, and we don't expect any blood clotting to happen. That's my opinion, and I wouldn't say like there will be TF present in bovine milk, but the way the milk preparation was not properly, maybe some steps are missed. That's my suggestion.
Yeah. Possible, I would say, but why should there be non-functional tissue factor in bovine milk? So if there is some kind of encrypted tissue factor, why should it be there, right?
Okay.
I mean, I would have also another comment.
Yeah, please go ahead, Carla.
Like if you, with 3000 G, you kind of destroy the EV corona, then, I mean, why you don't do it with the milk, with the human milk? Because they are treated the same way.
Yeah, I think that the tricky part is that bovine milk and human milk have really a very different composition with regard to the presence of casein particles and milk fat globules, etc. So I'm not sure whether you can easily directly apply a protocol that was developed for human milk to bovine milk. On the other hand, what I try to show you is that if you look at human milk, there is such an excess of tissue factor activity that I think even if bovine milk would not be treated optimally, I still think as long as it's not pasteurized, there should be such a high tissue factor activity. Just a few vesicles is already sufficient. And I really think it's not there. Also, the fact that bovine saliva quite potently triggers clotting, I think shows that the system also works for bovine fluids. So I really think it's unlikely, but you never know.
Thank you.
I posted the publication in the chat function, so you can just download it.
Okay, thank you.
Thank you, Johannes. And there's one more question. So I just want to ask, like, in case of hemophilia condition, were you able to check the combinational effect of tear, sweat, and blood together, or is just individual?
No, unless Johannes and Carla have done that secretly without me knowing it, but Perhaps they can answer this question.
I absolutely think so, that this is some kind of protective mechanism that was not really regarded previously. So it's quite interesting because I very seldomly see patients with a factor 7 deficiency. So they are lacking also factor 7 in the body fluids, and I already looked at this, and they have more this mucosal bleeding. So it's much more frequent in them. So I really think that they have these vesicles that bypass factor 8. Yeah, and that's interesting because there are also drugs, so-called bypassing agents, which are given to patients who have a factor 8 deficiency as a medication. And this, to me, it seems to be some kind of endogenous physiological factor 8 bypassing agents that just very directly and even faster trigger clotting. Yeah, so usually you need this amplification because in the vessel, if it clots too much, you have thrombosis or stroke or myocardial infarction. But this is not the case outside the vessel. So there can be a much stronger trigger without bad effects. Yeah.
Thank you. Ketnet, do you still have one more question? Ketnet?
I don't have any questions. Sorry.
Okay. Yeah, okay.
My part. Yeah.
It is raised.
So is there any more questions from the floor? If no, I would like to say thank you very much, Dr. Rienk Nieuwland, and all your collaborators for joining with us today. And we have prepared a certificate for your talk to say thank you from our end, and we'll send it to you in due course.
Okay.
Thank you very much
for the invitation, and also like to thank Johannes and Carla for being present and helping me out with these difficult questions.
Well, thanks, Rienk.
Thank you very much.
you.
Thank you.
Okay. Okay.
See you.
Have a nice weekend. Bye.